So today we're going to talk about DNA repair and mutations. So a mutation in DNA can lead to a loss of function, but can also give rise to genetic variation. So there are different types of mutations, including substitution, where a base is replaced, and there are two types. So it can be transition or transversion. So in transition, you'd have a purine, switch to a purine, and then in transversion, it would be like purine to a pyridine and vice versa. And then there's insertion, which is the addition of an extra base or extra bases, and then deletion, which is a removal of a base. And then there are different effects of mutation. So there's silent mutation, which is an alteration of amino acid does not occur, so it's synonymous. So even though there's a mutation, it can, for example, code for the same protein, so at the end of the day, the mutation would be silent. And then there's missense, where the sequence now codes for a different amino acid, and it would be non-synonymous. And then there is nonsense, which codes for premature stop codon. And then that, for example, is like doesn't make any sense because it stops prematurely, so nonsense. And then there's, of course, deletion, which is brought on by insertion or deletion, and it changes how the sequence is read, so it can shift the entire reading frame, resulting in a completely different structure. And they're all put into classifications, so there can be a neutral one where there's a change in amino acid, but the activity is the same, so essentially it's a neutral effect. There can also be loss of function, where now, like, for example, a protein that had this function no longer is able to work the same. There is a gain of function, which is the exact opposite. And then some can be lethal, and others can become conditional, where they work in one circumstance but not necessarily another. And then just for some background, de novo mutation are present in the parent's gametes, but they're not present in the somatic cells, so they're de novo, so they're essentially new. And there are different causes for mutation. So there can be spontaneous causes, so which is an error in DNA replication, recombination, and so forth. And then there's also, they can also be induced. So an example would be an exposure to a mutagen. And there could also be strand slippage. So for example, in strand slippage, you might have a section of the DNA that's read multiple times, which would result in insertion, and then there's some that aren't even read at all. And then spontaneous chemical changes. Under this would be depurination, which is the hydrolysis of a purine base. So this would affect adenine and guanine. And you also have deamination, which is the amine group changes to double bond O. And then UV light, which can induce thymine dimers. And of course, for all these mutations, there's DNA repair mechanisms. So the first one we'll discuss is mismatch. So this occurs shortly after replication, and it detects changes or like differences between the new and the old strand. And if there is a detection in like differences, then it can go ahead and begin the repair. And then there's base excision. So this works at the base level, and different proteins fix different mistakes. So for example, you wouldn't use a pencil eraser to erase the whiteboard. So different mistakes, different proteins. So an example, it would be for deamination. So cytosine going to a uracil, going back to a cytosine. I don't know if that makes sense. So C to U, which is the mutation, but changing it back into the proper C. So the protein would first remove the base, and then the AP endonuclease would come in, and it would cut it out, and then DNA polymerase would come in and fill in the space, resulting in a repaired DNA strand. And then we have the nucleotide excisions, and for the base, it literally works at the base level, but the nucleotide works at a much higher level, so it would scan the DNA for bulky damages, and one bulky damage would be thymine dimers, and it works at any time. So this is like a general overview of the mutations and the repair mechanism, and I hope this helps.